Light is something that can be turned on or off pretty easily, and in that way it's sort of like genes. Some genes should always be turned off and others should be turned off because they can cause things like genetic diseases. But mistakes can happen. Let's take our gene fairy. Sometimes she'll turn genes on that are actually supposed to be turned off. But what can we do then? Our gene fairy wishes that we could turn genes on or off with a switch, or that we could insert pieces of genes. One way to do this uses viruses. Now, normally when we think of viruses, we think of these bad creatures that actually harm us. But in reality, viruses are sort of the carriers for the DNA. And if there's bad DNA, they're bad. But if there's good DNA, they can be good. So how exactly can we use this to our advantage? So our normal virus has bad DNA, but if we change the DNA, it can become a good, useful virus. So let's use the example of hemophilia A. Hemophilia A is located on the F8 gene, and normally it's turned off. But in our patient Bob, it's actually turned on, and that's what's caused him to have hemophilia. Let's help our patient Bob. We'll start off by taking a bad virus, extracting the bad DNA, putting in the good DNA, making it a good virus, and then giving our good virus to a bad cell, which makes our new cell not good, but perfect. So now scientists will stimulate the cell to reproduce and undergo mitosis, put it into Bob's bloodstream, and voila, he is now cured. Phew, that was a ton of science and biology. But now, let's talk about some of the problems that can be associated with this virus vector solution. One problem is that the virus can't contain a lot of DNA. So that means that if we want to change out a ton of mutated bad DNA and genes, we actually have to put in a ton of DNA, which the virus can't hold. Another problem is that if the DNA isn't put into the right place, that means that the DNA can cause cancers or other genetic diseases that aren't directly related to the DNA in the nucleus. We have to make sure that the virus goes into the nucleus and inserts the DNA at the right position. And usually this will happen due to the natural tendencies of viruses, but sometimes it won't work. And that can lead to so many cancers. So this isn't a perfect solution, obviously. But it's helped to solve and cure so many genetic diseases that before there was no solution. But now, so that we can change the way medicine is impacted in our world, once and for all.